Hello, my name is André Voltzmann. I'm a group leader and lecturer here at the University of Hull at Biological Sciences, and I've been a researcher in molecular and cell biology for 17 years now. I am leading a Vice Chancellor's PhD scholarship that will focus on optimizing the manufacturing processes of viral vector-based gene therapies. Gene therapies are used to treat devastating diseases by addressing the underlying genetic defects. There are currently 6 gene therapies with market authorization that are based on a type of viral vector called AAV, with around another 100 in clinical trials. These AAVs are produced from cultured host cells. However, estimates indicate that with current manufacturing approaches, only 1 in 100,000 AAVs produced is able to successfully transfer the needed genetic material. This has led to somewhat of a bottleneck in the application of these therapies, causing their prices to be in the range of hundreds of thousands to several million pounds per dose, meaning we're not utilizing the full potential of these treatments. Unsurprisingly, this has resulted in inequalities in the access to these therapies. The overall aim of the research is to generate new knowledge about the biology of viral vector manufacturing, to make the process more efficient and gene therapies therefore more affordable and accessible for those who need them. The work has significant real-world impact and thus has potential for future industrial partnerships. This VC PhD scholarship project aims to provide new insights into the assembly and release mechanism of AAVs, which will allow us to make informed decisions on how to engineer cell lines and modify existing production processes to increase the percentage of functional AAVs. The first aim is to identify the cell factors used by AAVs for their assembly and release. The second aim is to study the role of these factors in these contexts. And the third aim is to generate a proof of concept that modifications of the identified pathways can be used to increase the amount of functional viral vectors. This project will be highly interdisciplinary and collaborative, and we'll be using a number of approaches to achieve the project aims, including AI-informed construct design, spatial proteomics, high-end microscopy, and various biochemical assays. This PhD scholarship project will be a collaboration between the universities of Hull and York. My lab will host the project and I will lead the supervisory team. I will be joined by Dr. Cheryl Walter, our resident virologist here at the Center for Biomedicine, and Professor Jeremy Mottram, who's the director of the York Biomedical Research Institute at the University of York. This collaboration is a fantastic opportunity to build a network with researchers in Hull and York by making use of regular seminar series, conferences, and career development opportunities at both universities. The PhD project will provide the opportunity to develop and hone relevant skills recognized as vital for the biotechnology and biomanufacturing industries, with a special focus on advanced therapy medicinal products. The knowledge and skills gained during the PhD will support many future career options, for instance, industrial research and development contexts. So this could be applied in cell line development and process engineering. Another option would be to pursue an academic research career. And of course, engaging with stakeholders and researchers at meetings and conferences would also train for a career option in scientific communication, public engagement, and public sector work on health-related subjects. If you're interested in this project, please apply, and don't hesitate to contact me at the address shown on the slide if you have any questions.